basic genetics لو field of medical genetics or basic genetics. في عندنا molecular وبiochemical physiological وcyto وبعدين oncogenetics or genetics of tumors. Oncogenetics is a branch of basic medical genetics concerning with studying the genetics of tumors, not only cancer but tumors in general. وفي major differences between benign or malignant tumors, لكن oncogenetics بتركز على الكانسر in particular. عشان نفهم برضو كل اللي هيجي بعد كده. Any activity in our cells is mediated through what we call metabolic networks. The examples of metabolic networks, a metabolic network is a pathway of too many components, mostly proteins, enzymes, and other cofactors, sometimes minerals or heavy metals. Any physiological function is mediated by one or more of the metabolic networks in our cells. The components of these networks are the products of our genes. Our genes control our life activities by synthesizing proteins and enzymes which form these metabolic networks and mediate the functions. Malignant transformation is a very mysterious and enigmatic phenomenon. إلى الآن we know too little about why cancer develops. نعرف too much about how it develops, but we don't know why cancer develops. لكن direct causes of cancer development, as we can see here, معلش لحظة. Oncogenetics or genetics of cancer comprises a study of genetic factors involved in carcinogenesis. These factors include proto-oncogenes. Proto-oncogenes دي هي mainstay of cancer development. The major factor in cancer development, regardless of other complementary factors, is conversion of a proto-oncogene to an oncogene. Oncogene is a gene which produces a protein cofactor or RNA molecule which has direct effect in converting the cell to cancer cell. Normal cell can be converted under the effect of the oncogenes and the oncoproteins or the proteins produced by oncogenes to cancer cells. The oncogenes come from mutations in what we call proto-oncogenes. What are the proto-oncogenes? Normally in our cells, there is a misnaming, a proto-oncogene. في misnaming لأن these proto-oncogenes are normal genes in the normal state which mediate important physiological and essential functions of the cells. Proto-oncogenes are genes which produce important proteins, which these proteins are essential for normal structure and function development and performance of the cell. They regulate the cell cycle, cell division, cell differentiation, and cell death as well. So the proto-oncogenes are vital genes. They are widely distributed in most regions of the human genome. This karyotype shows some of the proto-oncogenes in our cells. Each of these genes, written in red, is a proto-oncogene. As you can see, they are widely distributed in our genome. These proto-oncogenes produce or synthesize proteins. These proteins, as we say, they are essential for controlling cell cycle, cell division, cell differentiation, and cell death as well. When a proto-oncogene is mutated, mutation of an proto-oncogene either disrupt the gene becomes disrupted and out of order, or it may change the protein of the gene into an oncoprotein. So accordingly, when a proto-oncogene is changed by mutation to an oncogene, the cell begins an oncogenic process or transformation to malignancy. Not only this slide shows how cancer develops due to mutation-induced transformation of a proto-oncogene. We can see here, there is a proto-oncogene here on one of the chromosomes in the nucleus. Then a cancer-causing agent, we call it carcinogen. Carcinogen is a mutant factor which can cause cancer. When a cancer-causing gene or a carcinogen affects a proto-oncogene in a specific part of our chromosomes, then we get an activated oncogene. This activated oncogene begins transforming the cell to a cancer cell. This is, in general, how cancer develops due to mutations of proto-oncogenes. Proto-oncogenes are very important, and they are the mainstay of carcinogenesis, but they are not the only mainstay of carcinogenesis. Malignant transformations can start and develop by one of two main mechanisms: either mutation-induced transformation of proto-oncogenes to oncogenes, or mutation-induced disruption or inactivation of what we call tumor suppressor gene, and all later on. In addition to these two main mechanisms of malignant transformation, we have other complementary mechanisms which are needed for completion of the malignant transformation of cancer. What are the genetic factors involved in carcinogenesis? There are too many genetic factors involved in malignant transformation. أول حاجة is genome instability. For our cells to perform its physiological functions in a normal way, our genome must be stable. That's to say, our genes function normally and their products participate in forming the networks, metabolic networks in the cells, and metabolism goes normally. This is what we call the genomic instability. Genomic instability is disrupted in cancer by a major mechanism, which is a reactive stress replication, replicative stress leading to double-stranded breaks. Double-stranded breaks means breaks in both strands of the DNA. During replication under stress, we get what we call replicative stress. This replicative stress leads to disruption of the proofreading mechanism during cell division, and we get double-stranded breaks. Double-stranded breaks are the major factor which can cause genomic instability. This genomic instability usually affects a lot of our genetic material, and we get mass activation of dormant suppressed gene or mass suppression of active functioning genes, and reversion to early embryonic developmental stage, which is a characteristic feature of malignancy. A malignant cell, in contrast to the normal cell, is a non-specialized cell. It can do too many functions characteristic of the embryonic cells. بينما our differentiated cells only do the functions they are devoted for. Yani liver cells can do the functions of the liver, but malignant cells can do the functions of liver cells in addition to renal cells, probably other cells as well. Genomic instability is one factor involved in carcinogenesis, but in at a molecular level, we have other, or when we analyze the effects of double-strand breaks on our genomic stability, we get other factors. Tiny factor, DNA protective genes. We have genes which protect DNA against damage and against oxidative stress, for instance. DNA, these are different from the DNA repair genes. Most DNA protective genes are represented by what we call metallothionein genes. Metallothionein genes are genes which perform very important function in protecting our DNA against external and internal mutagenic exposure. So exposure to mutagens lead to damage of DNA or regulatory protective genes as well. So DNA protective genes are important for our DNA stability and performance. And when they get disrupted by mutations, this function is lost, and this paves the way to development of malignancy. After DNA protective genes, if they got disrupted or inactivated, we go to the proto-oncogenes. Proto-oncogenes are normal genes. They produce very important factors which regulate cell cycle, cell division, cell growth, cell development, cell death as well, or apoptosis. And when mutation affects these proto-oncogenes, they can either be disrupted and get out of order, or they may be transformed to oncogenes. Once a proto-oncogene is transformed to oncogene, what can happen? We get enhanced expression of the oncogene products, increased synthesis of the oncoproteins, transcription factors, coding and non-coding RNA. All these factors are essential for malignant transformation. Then we get third genetic element or factor involved in carcinogenesis, which is tumor suppression genes. Tumor suppression genes are genes which regulate too many functions in our cells. When these tumor suppression genes are inactivated by mutation-induced disruption or chromosomal deletion of these tumor suppression genes, P53 gene is the most important of these genes. Cancer can develop and tumor can develop. Malignant transformation of the cell can happen as well. Then we get DNA repair genes. As we know, point mutations happen continuously during the cell division. And it is estimated that in a normal cell cycle of 20 hours, if a cell is going to divide within 20 hours, nearly 12,000 point mutations happen during these 20 hours. متخيلين ان في كل سيل سايكل بتاخد 20 ساعة مثلا 12,000 point mutations happen. لكن the majority of these point mutations are corrected by our DNA repair system. Our DNA repair system is a very strong and very resilient system. It can detect point mutations by the proofreading system. Proofreading system ده عبارة عن enzymes تتأكد in new strand being formed عشان نعمل new chromosome و new cell later on. A new strand is exactly complementary to the other new strand. Any point mutation detected is excised and normal nucleotide is inserted. فده البروفريدنج سيستم in short. فال DNA repair system is very important for correcting the huge number of point mutations which develop during each cell cycle. بالتالي mutation induced disruption or an activation or deletion of these DNA repair genes and double strand break repair, these are very important factors in development of cancer and carcinogenesis. Then we get to what we call apoptosis regulatory genes. As we know, apoptosis is a programmed cell death. This is the definition of apoptosis. Apoptosis is a very important protective mechanism. Why? Because with time, mutations accumulate in our DNA, both mitochondrial DNA and nuclear DNA. And with time, if these mutations are allowed to stay for longer times, they can convert some of the proto-oncogenes into oncogenes. طبعا mutations can cause cell death, no problem. Other cells will replace the dead cells. لكن المشكلة إن with accumulation of mutations, some of these mutations can drive the cell into malignant course and cancer develops. So apoptosis of heavily laden cells with mutations is a protective mechanism against cancer. لو اتسابوا mutation, cell might get cancer. Like if the cell goes through apoptosis pathway, it will die, and so we will obviate the development of cancer in the cell. So apoptosis regulatory genes are very important. Not only they are most important بالنسبة للcancer, لكن they are essential during development. لما بيحصل hollow organs, لما بيحصل cavitation, لما بيحصل folding during embryonic fetal stages of development, apoptosis is essential. لو apoptosis ما بيحصلش, we get a huge list of congenital malformations in the developing fetus. So apoptosis as regards cancer is important because it protects the cell from being driven into a malignant course. فده fourth or fifth genetic factor involved in carcinogenesis. Then we get what we call metastasis enhancing genes. المشكلة الكبرى في الكانسر هو الميتاستاسيس. If metastasis can be challenged and arrested in cancer, cancer would be a local lesion that can be treated by conventional methods. الهورر of cancer أساسا كله في metastasis to distant organs, and this is the final cause of death of patients اللي هو metastasis. سامعيني يا جماعة ولا لا؟ سامعيني ولا مش سامعيني؟
تمام يا دكتور.
سامعين؟ طيب. فالميتاستاسيس هو
سامعين حضرتك كده.
الورست والهورrible fate of cancer هو due to metastasis to distant organs and destruction of these organs. Metastasis enhancing genes, we get a very important group of genes, what we call matrix metalloproteinases. As the term implies, they are enzymatic in nature. These matrix metalloproteinases play important roles in forming the architecture of the tissues. Tissues are, each tissue has a specific architecture, structural architecture, and this architecture is determined by the matrix proteins in between the cells. Most of these proteinases are enzymes, collagenases, gelatinases, and streptolysine-like compounds, stromycin genes, materilysine genes, etc. These genes are very important, and they represent the pivotal factor in metastasis, because when cancer develops, these genes, under the effect of the oncoproteins, pave the way for spread of cancer to neighboring tissues and then to distal organs as well. So degradation of the extracellular matrix proteins under the effect of these metastasis-enhancing genes leads to invasion of neighboring tissues and metastasis of malignant cells to distant organs. We have what we call anti-metastatic genes. Anti-metastatic genes are genes which can arrest or decrease the ability of malignant cells to break intercellular barriers and invasion of neighboring distant sites. These anti-metastatic genes, many of them, like a major function of them, they produce or synthesize proteins which are essential for what we call cell contact. Cell contact for a tissue to get its final architecture and strength. The cells, we have cell contact points عبارة عن proteins fibrillar protein mostly بتربط cells together. These are example of what the anti-metastatic genes produce. They produce the cell contact proteins. And one important feature of cancer is loss of contact inhibition. What is contact inhibition? When cells are contact to each other, then their division is limited by the capacity, what we call the capacity of the architecture of the tissue. يعني التيشو موجود بحيث ان عشان التيشو ده stays maintain structure normal, there is a specific number of cells and intercellular materials that form this tissue because the cells are in contact to each other. If this contact is inhibited, what we call contact inhibition, which is a characteristic of cancer, the cells can divide haphazardly and they do not respect their normal program of tissue structure. This is a function of the anti-metastatic genes. But in general, we have too many genetic factors which are involved in carcinogenesis. We have the proto-oncogenes, the tumor suppressor genes, DNA protective genes, DNA repair genes, apoptosis regulatory genes, metastasis and anti-metastasis genes, and lastly, genome instability. Those genome instability can cause double-strand breaks in too many areas of the genome on the chromosomes, and if these breaks happen in sites of proto-oncogenes, they might induce the proto-oncogenes, convert them to oncogenes, and a malignant tumor begins to develop. So these are the genetic factors involved. في طبعا too many other factors in details, لكن these are the major or main genetic factors involved in carcinogenesis. في أي سؤال على اللي قلناه لحد دلوقتي؟ في أي سؤال يا جماعة؟
لا تمام يا دكتور، مفهوم حضرتك.
واضح؟ طيب يبقى ده genetic factors involved in carcinogenesis. How بقى development of malignant tumors? The formation of malignant tumors or carcinogenesis is genetically determined. It is a complex, progressive, multi-stage process. Complex لأن فيه too many genetic elements are involved in the process. Progressive because cancer cells always have selective advantages over normal cells. A normal cell is overcome by cancer cell as regards the potential to grow. A survivor, a cancer cell can grow more efficiently than a normal cell. And because of apoptosis is suppressed in cancer cells, they can have longer survival time. Also, They can, for instance, get less nutrients through glycolysis, and in spite of this, they can grow and multiply and spread. So it is a progressive because of the selective advantages of cancer cells over normal cells. And it's a multi-stage process. Multi-stage process is this is a characteristic of cancer, لكن up till now it is still a mysterious. يعني مثلا radiation induced cancer ممكن develop after 10, 15 or 20 years. And the mutation happens upon exposure to the radiation, لكن the malignant process begins to start after a very long time, many years. We don't know why يعني multi-stage process في explanations, لكن these are details, لكن the important characteristic of malignancy is it is a complex, progressive multi-stage process, and as we said, primarily caused and starts by either one of two main mechanisms. A mutation-induced transformation of proto-oncogenes to oncogenes. وخدوا بالكم البروتو-oncogenes, as we said, is a misnaming. لأن في الأول كان الأونكوجين بنعرف it was another gene. This the father gene or the other gene was called the proto-oncogene. After that, it was discovered that most proto-oncogenes are genes essential for normal function of the cell. لكن التسمية فضلت، لكن دائما نعرفوا إنهم this is a misnaming. Proto-oncogenes normally in our cells we have too many proto-oncogenes, and they are essential for performing cellular functions. The second major mechanism, mutation-induced disruption or inactivation of tumor suppressor genes. In addition to these two main mechanisms, في complementary mechanisms which participate in tumor development to tumor growth or invasion of neighboring tissue metastasis. As we said, مثلا طيب دي معلش اقولها لكم عشان يمكن الديفينيشنز. البروتو-oncogenes they have the characteristic of the three major categories of genes. احنا genes بنقسمهم in general to three major categories because or in view of their functional characteristics. We have master genes, structural genes, and regulatory genes. Master genes or بنسميهم أحيانا تلاقوهم housekeeping genes. These genes function all over the clock من ساعة ما الزيجوت is formed till death of the cell. They are essential for normal cell function all through the lifespan of the cell. Master genes examples include مثلا genes which are responsible for the cell cycle and cell division, because this is a continuous process. Genes which are responsible for ATP production in the cytoplasm, because without ATP the cell cannot live or cannot perform any function. Transmembrane transport genes. A cell is not a solid structure. A cell is على طول we have molecules going into the cytoplasm and metabolites going out of the cytoplasm. If these transport mechanism or transport feature of the cell is interrupted in any way, the cell can die within very short time. يعني water inhibition by the cell of sodium, potassium, or calcium, if transport of any of these molecules across the cell membrane is disturbed, in few minutes, the cell can die. These are examples of master genes, genes which function all through the cell life. But then we have structural genes. Structural genes are genes which produce specific proteins or enzymes for specific physiological cellular function. زي مثلا insulin gene. Insulin gene is a gene which produces insulin when we have hyperglycemia after food, and when insulin lowers the glucose level to euglycemic stage, then the insulin gene stops function. This is an example of structural gene. Structural gene is a gene which produces a protein or an enzyme which is essential for mediating one or more cell function. Then we get regulatory genes. Regulatory genes are genes which regulate the function of structural genes. And mostly they synthesize protein factors and small coding, short coding, and long coding RNAs, which regulate the huge number of networks in our cells. تقريبا in our cells في حوالي 4,100 networks. Networks دول composed of proteins and enzymes and heavy metals, and other small factors. All these metabolic networks function under the effect of proteins and enzymes produced by regulatory genes. So this is in general the types of genes in our cells. Proto-oncogenes have the characteristics of these three major categories of genes. They are master genes. They function around the clock. من أول ما الزاجل بتتكون till the cell dies. Oncogenes are structural genes because they responsible for synthesis of proteins, RNA. Well, proto-oncogenes are regulatory genes as well because they, through their protein or RNA products, they regulate cellular processes programmed and maintained under the supervision of these genes. Mutations of proto-oncogenes sometimes turn them to oncogenes. The oncogene exhibit what? A major difference between proto-oncogenes and oncogenes is a quantitative difference as regards gene expression. Oncogenes exhibit increased production of these proteins produced by the proto-oncogenes. This leads to increased rate of cell division, decreased differentiation capabilities of the cells, inhibition of cell death, loss of contact inhibition. These are some of the features of what we call the malignant phenotype. Malignant phenotype يعني characteristics of the cancer cell which differentiates it from normal cell. There are many features which characterize the cancer cell from normal cells. These differences are referred to as the malignant phenotype. وزي ما قلنا increased rate of cell division with decreased ability of differentiation, loss of contact inhibition, arrest of apoptosis. All these are examples of what we call the malignant phenotype. Molecular mechanism of carcinogenesis as regards mutation-induced transformation of proto-oncogenes to oncogenes includes these four major molecular mechanisms. First, as we can see here, if we have here a proto-oncogene, then we might get point mutation within the control element of the gene and the promoter of the gene, or point mutation within the gene itself, or chromosomal abnormalities leading to gene amplification. And instead of having two copies مثلا of a proto-oncogene, we might have multiple copies, or translocation of proto-oncogene to another locus in the genome If this locus is controlled by a stronger promoter, the proto-oncogene might get under synthetic stress and it produces more of its proteins. All these consequences of these four major molecular mechanisms of proto-oncogene to oncogene transformation will lead to increased or excess synthesis of these proteins. As we can see here, instead of normal growing or normal growth stimulating protein, we get this protein but in excess, leading to increased cell growth and cell division. Also, gene amplification, we will get excess protein. Point mutations, if these mutations do not disrupt the gene or inactivate it, they might transform it into an oncogene with the result of synthesis of its proteins in excess. All these mechanisms will lead finally to increased rate of cell division, increased cell growth, increased ability of the cell to invade, inhibition of contact contact inhibition between cells, leading to facilitation of spread of cancer gene to neighboring tissues and to distant organs as well. ده بيلخص برضه المولكيولر mechanism point mutations leading to synthesis of new proteins with oncogenic activity, point mutations leading to overproduction of protein products which increase all the cell functions regularly under the control of proto-oncogenes, gene duplication or amplification due to chromosomal abnormality events, or chromosomal translocation. Chromosomal translocation can relocate a proto-oncogene to a new chromosomal site. In this new chromosomal site, the proto-oncogene might lose its function. And this is one mechanism of why chromosomal translocation can cause disease in general away from cancer. Proto-oncogene performs and mediates important critical cellular functions. In a chromosomal translocation mechanism, a proto-oncogene might be relocated to a new site and it might lose its function on this site, and all the cellular functions dependent on the protein of this proto-oncogene might be lost, and this is one aspect of chromosomal abnormalities, functional pathological functions of physiological processes which occur due to chromosomal translocations. Or on the other hand, this proto-oncogene is relocated into a site downstream of an overexpressive promoter or enhancer site, higher expression of the translocated proto-oncogene and oversynthesis of its products can occur. واضحة النقطة دي يا جماعة؟
واضحة يا
واضح
دكتور
ان
حضرتك.
الكروموزوم translocation can cause can pave the way for some cancers if a proto-oncogene is relocated into a site downstream of an overexpressive promoter. فيؤدي higher expression oversynthesis of its products, of its oncoproteins, and this also paved the way for development of cancer. Or another mechanism which is triggered by chromosomal translocation and can be involved in carcinogenesis is formation of new fusion sequence. بمعنى proto-oncogene might be relocated to another site. If during this translocation part of the oncogene gets fused with preceding or other sequence of the DNA, it might form new fusion sequence. This new fusion sequence is a new part of DNA. Then a part of the proto-oncogene has been inserted. We can put as a new insertional mutation in this part. This new fusion sequence sometimes might synthesize fusion proteins with oncogenic activity. And this is responsible for the phenomenon of finding new proteins in cancer. In cancer, we get normal proteins in higher levels, but sometimes we get new proteins, oncoproteins, which are not part of the normal human proteome. Normal human proteome, like the normal human genome, has been defined. We know all the normal proteins in our cells as we know all the normal genes in our cells. But in cancer cells, sometimes we get confronted with strange proteins which have not been detected before in our proteome, and this probably express the new fusion sequence and new fusion protein with oncogenic activity that might develop in cancer. That explanation of the unexpected proteins in the blood of cancer patients or in their cells due to this specific type of chromosome translocation. But then these are the major factors which transform proto-oncogene to an oncogene, but there are complementary molecular mechanisms which also help in cancer development, and these complementary mechanisms which represent the worst expression of cancer, لأنه some of these complementary mechanisms involve enhancement of metastasis and spread of tumor. هنقولها دلوقتي mutation induce interruption of genome stability زي ما قلنا, mutations leading to disruption, inactivation, deletion of genes responsible for protection of DNA زي ما قلنا against heavy metal toxicity and oxidative stress, هما metallothionein genes. Or mutations leading to also disruption, inactivation, deletion of genes that regulate apoptosis. These mutations cause deficiency of apoptosis initiation, completion enzymes and proteins, which constitute essential mediator in apoptosis pathways. Pathogenetic arrest of apoptosis results in important quantitative phenotypic features manifested as marked increase in cell number and progressive increase in tumor mass. When tumor cells, apoptosis is arrested within tumor cells as a major feature of the malignant phenotype, what will happen? We will get increase in cell numbers and progressive increase in tumor mass. This increasive arrest of apoptosis or slowing down its predefined rates imparts a selective longer lifespan advantage of cancer cells. So for normal cells, they begin to go through apoptosis after a specific number of cell divisions, but for cancer cells, they can reach this fate after many folds increase compared to the normal cell rate. فالكانسر cell بتقدر تعيش for very longer time. بنقول immortality, it is not immortal لكن بتعيش for very long lifespan compared to normal cells. فالarrest of apoptosis بيدي this very important selective advantage of cancer cell over normal cells. بعدين very important also positive weight for weight balance in favor in cancer cells. بمعنى إن لو بدأنا بnormal cells after some time هنلاقيهم وصلوا to specific number of cells and specific weight. For malignant cells, they can cross this limit by many folds. فده positive weight for weight balance. The positive weight for weight balance as regards cancer cells has an important direct impact on enhanced survival of the tumor لأن tumor size لو افترضنا ان at a specific organ we have normal cells and neighboring tumor cells, tumor cells much larger as regards weight for weight, so they can get more food, more blood, more oxygen compared to the neighboring normal cells without بديهم selective advantage as regards survival and growth as well. وده is an important feature for tumor development with decreased rates of apoptosis, the cardinal feature of the malignant phenotype of most malignant tumors. Type. In addition to mutations of apoptosis genes, mutations of induced overexpression of metastasis genes. If these metastasis genes are mutated in a way that leads to their overexpression, then they will produce more proteins with enzymatic capabilities, collagenesis and stromlysin والأذر يعني حاجات كتيرة قوي يعني بس we have to know يعني the meaning والexample زي بتتحفظ يعني مش ضروري. فال overexpression metastasis genes pave the way for invasion of neighboring tissues, neighboring cells, and distant metastases. The protein products of these genes, which are we call metalloproteinases genes, have important regulatory functions in maintaining the normal condition. Normally, they have important regulatory functions in maintaining the extracellular environment, intercellular contact, and local communication and cooperation pathways between cells and tissues for safeguarding and maintaining the local tissue architecture. فالmetalloproteinases are very important in the normal state to maintain and protect the structural architecture of our tissues. An intact local tissue architecture is a vital and essential anti-metastasis defense mechanism. This is the significance of mutations in these metalloproteinases. When metalloproteinase genes get mutated, then they lose some of their functions. They lose this ability to keep intact local tissue architecture. And this is important for paving the way for spread and metastasis of malignant cell. عشان كده this one of the important complementary mechanism in carcinogenesis, which plays a central role in enhancing tumor spread and metastasis. Metastasis is the most drastic consequence and devastating stage of cancer progression. زي ما بنقول, without metastasis, cancer would be just a local lesion, local pathology, which can be dealt with by surgery, local irradiation, local chemotherapy injection, etc. المشكلة كلها in cancer in metastasis, all drastic consequence and the cause of death in all cancer patients finally. It underlies and dictates the final fate of cancer patients, and without metastasis, cancer would have been a local and localized pathology amenable to total cure by conventional treatment modalities. وعشان كده النقطة دي مهمة قوي وللأسف إلى الآن ما فيش اهتمام كافي بيها, لأن though researches aiming at disclosing the still ambiguous and vague mechanisms that regulate tumor spread and metastasis, this research represents the actual true rational and logical therapeutic approaches for controlling and curing cancer. لأن لو في research aiming at arresting of or challenging metastasis, this is rational or logic therapeutic approach to control and cure cancer. But unfortunately and unregrettably also, they are the least in priority among researchers aiming at finding palliative therapeutic approaches for cancer. لو بصيتوا على الانترنت بتشوفوا الكانسر ثيرابي ريسيرشز هتلاقيها يمكن مؤخرا معظمها بقى موجه للاميونوثيرابي اوف كانسر. وذيس از نونسنس. ان الكانسر ما هوش ديزيز. الكانسر is a biological system. Cancer is not a disease. Cancer is a biological system parallel للnormal life system in our cells. وإلى الآن كل حاجة بتحصل الكانسر cells can overcome it because it is not a disease, not a dead cell which tries to infect the body. لا, ده الكانسر cell system has its main objectives of creating another system of it. لكن في لو في آخر المحاضرة دي in short، لكن لما تدخلوا على السايت يا ريت تنزلوا الارتكل اللي هي كانسر some genetic considerations، هتلاقوا فيها more بس theoretical discussion، لكن يعني in detail عن ان الكانسر is another biological system, it is not just a disease. وعشان كده الى الان معظم الترايلز to combat cancer are big failure للاسف الشديد. ما علينا يعني. طيب طبعا قلنا في tumor suppressor genes are an important category of genes which suppress development of malignancy in the normal condition. Mutations of these tumor suppressor genes are associated with development of cancer. وفي specific correlations between specific of these tumor suppressor genes and specific types of malignancy. For instance, B53 gene mutations are associated with development of brain tumors, carcinoma of معظم الحاجات دي وساركوما لوكيميا etc. مثلا breast cancer 1 breast cancer 2 مع breast cancer ovarian carcinoma. مثلا retinoblastoma tumor suppressor gene زي ما قلنا خدوا بالكم الميس نيمينج، احنا اللي بنسميه retinoblastoma gene، لكن هو gene is very important لانه synthesize a specific protein called E2 protein which is essential for regulating normal cell cycle. فretinoblastoma gene mutation of this gene results in development of retinoblastoma, sarcoma, carcinomas of hereditary bladder, breast and lung, etc. So these are just examples of some of the tumor suppressor genes and tumors which result due to mutations of these genes as well. الأونكوجينز يعني زي ما قلنا البروتو أونكوجينز are important. They produce growth factors and growth factors receptors, proteins that affect apoptosis. They produce proteins that control cell cycle, dicycline, and proteins which regulate apoptosis of the mitochondria. They produce transcription factors, produce protein kinases which activate. These are very important regulatory proteins and enzymes in our cells. Mutations of these proto-oncogenes ممكن نتخيل they disturb cell cycle, cell division, they disturb apoptosis, they disturb all metabolic networks in our cells, and these are all of these are just few examples of what we call the malignant phenotype, which is imparted to the cell under the effect of oncogenes. الoncogenes of human genome, too many برضه. ده table just example of the oncogenes and their functions and the relevant tumors which develop or arise due to mutations affecting these proto-oncogenes and forming يعني مثلا growth factors وميتوجينز السيس cytoplasmic cis proto-oncogene ده is respond أو belonging to the category of genes responsible for cell growth and mitogens. Its gene function, it induces the cell for proliferation. If mutations affect this proto-oncogene, we might get these tumors. And epidermal growth factor receptor, this is a very important proto-oncogene. It produces the epidermal growth factor receptor. It is important for signal transduction. Signal transduction ده زي ما تقولوا communication system inside our cytoplasm. But يعني لو جت مرة نبقى نقول signal transduction لأن ده major regulatory mechanism which cooperates our pathways, metabolic pathways in our cytoplasm. Many proto-oncogenes produce proteins which regulate this signal transducing function, and mutation of these proto-oncogenes results in development of these examples of tumors. الحاجات دي كلها يعني حفظ يعني ما فيهاش فهم طبعا. بتعرف البروتو-oncogene, its function, and the tumors which develop due to or second to mutations affecting these proto-oncogenes. ده برضو يعني ما فيش جديد يعني how normal cells proto-oncogene is transformed to oncogenes or tumor suppressor genes if they are suppressed or inactivated by mutations. Then we will get disturbance of all these important functions of the normal cell, signaling enzyme, cell proliferation, transcription factors, all these growth factors, all these when they get interrupted or disturbed, then malignant transformation of the cell might begin. الفانكشن aspect of carcinogenesis ده برضو بيبين some of the characteristics of the malignant phenotype. The malignant cell, as we can see here, the malignant cell, we get loss of contact inhibition. Contact inhibition that is very important biological phenomena. Biological phenomena وكان discovered first time يعني في اليست cells لما لو ان اليست colonies only بتيجي عند specific size and stop division and growth. This is due to what we call cell contact effect. Cell contact effect ده عشان يحفظ perfect nutritional status for the cell colony بالنسبة لليست مثلا. بعدين اكتشفنا انها is a very important characteristic selective advantage for cancer cells. Cancer cells, they lose the contact inhibition characteristic of normal cell, وبالتالي they can divide more than the local tissue can tolerate, and they begin, this also is an important factor in beginning to disrupt the local architecture and spread to and invasion of neighboring tissues. الفيتشرز دي زي ما بنقول عشان كده الكانسر is not a disease. لما بيحصل bacterial infection البكتيريا ما بتقدرش تعمل colonies in the neighboring ولا invade the neighboring cell. فعشان كده these are only characteristics. These are specific features of malignant cells as well. فنلاقي فيه loss of contact inhibition, increase in growth factor secretion, increase in oncogene expression, loss of tumor suppressor genes. This is in contrast to the normal cells where oncogene expression doesn't happen or rare, intermittent or coordinated growth factor secretion, presence of tumor suppressor genes, and DNA protective genes, DNA repair genes, apoptosis pathway genes, all are in normal status. المalignant phenotype هم structural features which characterize structural and functional properties that constitute the malignant phenotype. The major characteristics of the malignant cells play induced or spontaneous mutation of specific oncogenes, defective repair mechanisms of mutated genes due to defect in DNA repair enzymes, loss of control of cell cycle, leading to unrestrained cell division and hyperplasia, altered pathways of apoptosis resulting in immortality or longer lifespan of malignant cells, modification of intercellular connections and adhesion molecules, leading to loss of contact inhibition mechanism, induction of angiogenesis. This is an important characteristic of malignant cells, induction of angiogenesis. Why angiogenesis is important for cells? Because if a cell mass grows to a critical level, then the central portion of this cell will die because no blood will reach this central portion of the tumor. So angiogenesis is a specific characteristic feature and essential feature for survival of tumors. Well, angiogenesis is a feature which characterizes the embryonic and fetal stage of life. During fetal stage, cells, genes which can create new vessels, new blood vessels, so this is angiogenesis, that's creation of new capillary and blood circulation systems. This angiogenesis is characteristics of the embryonic and fetal life. Malignant cells regain these fetal characteristics. They can induce angiogenesis. Normal cells cannot do that. Normal cells cannot do that unless after injury for the liver. Liver لما بيحصل فيها injury وبنشيل الحاجة can induce angiogenesis. Otherwise, no other parts of the body can induce angiogenesis, which is the characteristics of the fetal stage of life without a reversion to the embryonic or fetal stage. This is the characteristics of malignant cells and is an expression of genome instability. Genome instability in cancer, بنلاقي mass activation of dormant genes، بمعنى إن مثلا الجين التيومر في الليفر can metastasize to the lung and begin forming lung tissue, begin forming synthesis of corticosteroids, or it begins synthesis of any immune components in it. Normally dormant cells, normally dormant genes في differentiated cells get out of restriction في malignant cells. نلاقي mass reactivation of dormant cells, or on the contrary, mass inactivation of functioning cells. These are examples and consequences of genome instability on malignant cells. Also, in addition, we have increased dependence on glycolysis. Glycolysis is a metabolic pathway which produces a very small amount of ATP. For each 38 molecules of ATP produced by oxidation, normally, oxidative phosphorylation, we get only 2 ATP molecules from glycolysis or oxidation of sugar through this glycolysis. But tumor cells بالذات عشان تدي energy for the inner mass of tumor cells in the depth of the tumor, they can regain glycolysis as well, which happens in early embryogenesis, but is quickly lost, promptly lost with phytogenesis and with ripening of the ATP phosphorylation system. Increased dependence on glycolysis is a characteristic of cancer cells. Epithelial and mesenchymal transition of cancer cells. This is due to bardogenome instability affecting genes. لأن الفرق بين epithelial وmesenchymal are genes inactive here, active here, or vice versa, and invasion of surrounding tissues and metastasis to distant sites. الفانكشنال aspects احنا قلنا affection of cell cycle of apoptosis, angiogenesis, invasion and metastasis, والmalignant phenotype زي ما قلنا they are characteristics of the malignant phenotype of malignant cell. There is self-sufficiency. They depend on their self for producing the whole of the proteins they need. Insensitivity to growth inhibitory signals. And زي ما قلنا, growth is controlled by promoting and by inhibiting factors. Cancer cells lose their sensitivity. They do not obey the orders of growth regulation. لأن في loss of contact inhibition and في internal fences of growth factors in excess of what the inhibitory signals can tolerate. Invasion of apoptosis. Apoptosis is decreased or arrested in cancer cells. Limitless replicative potential. They replicate at a very high rates compared to normal cells. They can create new blood circulation system within their mass, or angiogenesis, or invasion and metastasis, with effect in DNA repair, loss of DNA repair genes due to mutations is also characteristics of too many tumors. And even when the DNA repair system is functions, it cannot challenge the capability of cancer cells to produce too many factors, which can induce two more mutations that the DNA repair system cannot cope with. الكريتيكال دواء summary of we said, يعني ما فيهوش حاجة جديدة. Imprinting defects in carcinogenesis. احنا عارفين imprinting is change in gene expression ability. This is simply what imprinting means. Imprinting, we have three major types of imprinting. We have special imprinting when genes in neighborhood of specific proteins might be activated or inactivated. That is very characteristic of plant cell imprinting. We have temporal imprinting. This is the important type of imprinting which happens in cancer. يعني ايه temporal imprinting? This means imprinting related to the stage of development of the cell. يعني time dependent imprinting. As we know, in early embryogenesis, with beginning of differentiation of the cells, all the genes in a cell, in a specific differentiated cells, which produce proteins which are not needed for the cell function, get imprinted or inactivated. بمعنى الليفر سيل مثلا، الليفر سيل فيها نفس السيلز اللي موجودة في الأديبوز سيل اللي في البرين سيل، in liver cell, in muscle cell. Any cell, any somatic cell in the body has the same complement of chromosomes and genes like all other cells in our body. A difference happens due to what we call selective imprinting. بمعنى مثلا الاكزامبل بتاعنا الليفر سيلز لو افترضنا ان في 4,000 or 38,000 genes in the cell, all these are inactivated or imprinted with the exception of the genes needed for the liver cell to perform its functions. The liver cell produces مثلا liver enzymes, liver proteins, etc. Only the genes which are needed for the liver cell to perform its functions are activated, they are active. All other genes are inactivated. The same applies also to any other organ. For instance, in renal cells, the kidney cell, only within the kidney cell, all the genes necessary for performing functions needed by this specific gene, whether it is in the cortex or in the medullary system or any way, are active. All other genes are inactive. In cancer, this selective temporal imprinting is lost. بمعنى إن بنلاقي إن في الكانسر سيل مثلا لو في liver cancer بنلاقي إن all the inactivated or temporarily imprinted genes in the liver cell can be reactivated. بمعنى نلاقي الهيباتوما is capable of synthesis of مثلا corticosteroid hormones or growth hormone or components which are not normally expected to be synthesized by the liver cells. ده اللي بنسميه loss of imprinting. فالimprinting means inactive normally يعني in very simple way. Imprinting means that all the genes in a cell which are not needed for mediating these cell functions are inactivated. Inactivation ده بيحصل very early during embryogenesis and phytogenesis as a part of the cell differentiation process. Cell differentiates, all the genes it doesn't need are inactivated, only the genes which it needs for its functions are activated. Loss of this temporal imprinting is a major characteristic of cancer cell, وهو اللي بيفسر why cancer cells can do all the functions of all other body cells. And this is why they are self-dependent. They do not depend on other cells to get their needs for survival, for growth, and for division. الموليكولار تيومر ميتاسيس زي ما قلنا تيومر ميتاسيس اهم حاجه ان الماتريكس metalloproteinase mutations of these genes pave the way for malignancy to invade neighboring tissues and metastasize to distant organs. Where this selective genomic reactivation or suppression of genes معلش أنا هقراها يعني عشان تبقى واضحة من الأول. Although the enigmatic phenomenon of tumor metastasis is responsible for the lethality of the vast majority of tumors, the nature of the pathogenetic mechanism underlying and mediating metastasis of malignant cells are still poorly understood. اللي احنا عارفينها بس اللي هو the role of metalloproteinases in disintegrating the extracellular environment of the tumor so the tumor cells can move freely and go away from its mother cells. Selective genomic reactivation and or suppression of genes responsible for regulating cell movement, such skeletal modifications and intercellular adhesions responsible for cell contact, resulting in reversion to the early embryonic fetal stage. لأن الحاجات دي كلها, all these are features which characterize the early embryonic fetal stage, where cell migration to and localization in distant organs plays a crucial role in organogenesis. زي ما احنا عارفين السيل migration is an essential stage of development. لو ما فيش cell migration too many diseases can develop في البرين يعني in particular وفي الليفر وفي الكيدنيز. فالcell migration is a characteristic of embryonic fetal stage, and they are a characteristic feature of cancer cells as well. Metastasis can be considered as a phenomenon which equal to or similar to cell migration. Cell migration happens in the fetus, and metastasis happens in malignancy in body organs. يعني برضو الماتريكس metalloproteinases, these are genes, they have important physiological functions. يا ريت تبقوا تقروها في الفسيولوجي يا جماعه، لان زي ما انتم شايفين هنا الفسيولوجيكال roles of these metalloproteinases are important في too many functions. مثال ذلك البون ريموديلنج مثلا، لان الكولاجينيزس، لان اي حاجه من الديفلوبمنت structural architectural formation of organs or tissues بيحتاج modeling by positive addition and negative deletion of components. الماتريكس metalloproteinases are important في aspect of deletion of components. زي في growth factors هي بتجيب الجروث هرمون مثلا مع والثايروكسين بيلعبوا important roles في الاديشن aspect of organ development. الماتريكس metalloproteinases بيلعبوا الديليشن aspect of organ development. فقريت بتقروها في الفسيولوجي in detail شويه الفانكشنز of matrix metalloproteinases. ادي احنا قلنا يعني mostly they are proteins with enzymatic catalytic abilities which can destroy the matrix fibrillar proteins والmatrix proteins around the اللي هم بيمثلوا physical normal barriers to malignant spread. فبالتالي when the function of these metalloproteinases are in excess due to mutations in the genes, they destroy the around the tumor barriers and so help in invasion and metastasis of the tumor to distant tissues and organs. يعني دي كلها حاجات يعني شرح للي بنقوله. والانتي ميتاسيس برضه زي ما قلنا. الانتي ميتاسيس طبعا ما هماش مقتصره على الميتاسيس سبريسور جينز اونلي، دي اكزامبلز. مثلا الكاد 1 جين ده is an inhibitor of metalloproteinases. The arrest metastasis include gene that include synthesis of tissue inhibitors or metalloproteinases. The host immune responses also are anti-metastasis mechanism. As we know, يعني لما بنشوف الهيستوباثولوجي of cancers, most cancers في the early stages at least بنلاقي they are surrounded by many lines of immune system of B lymphocytes, T lymphocytes, killer system, phagocytes, etc. This is an anti-metastasis mechanism, but it is not genetically regulated, but it represents the response of the body to any foreign body inside it, اللي هو immune response. فدوا is an anti-metastasis mechanism, but it is not genetically determined, but rather physiologically determined response of the host to existing foreign bodies. Then some of the oncoproteins have different structure than the normal proteins. The protein produced by the proto-oncogene might get changed when it is produced, might appear as an abnormal or foreign protein to the immune system, and so it begins to surround, try to attack, challenge, or define this resulting tumor. فالhost immune response is an anti-metastasis mechanism as well. Then oncostatic and anti-metastatic role of melatonin. الميلاتونين في الحقيقة الهرمون melatonin has very important functions in many aspects of the body. Is a major regulator of the response to light and dim or day and night changes in our circadian rhythm or circadian biological is a regulator of this. لقينا الميلاتونين كمان ليه important functions. مثلا it protects the cell against radiation induced chromosome breakage. It inhibition of tumor development induced by chemicals carcinogens in animals. Melatonin also exhibits a variety of oncostatic properties and metastasis suppressing and arresting effects. We don't know how, لكن it might do so through its effect in modulating some of the metabolic networks in our cytoplasm. In the anti-mutagenic or anti-clastogenic functions of melatonin, anti-clastogenic يعني it can protect against chromosome breakage which is induced by radiation. The meaning of anti-clastogenic. Clastogenesis is chromosome breakage. Melatonin has many important physiological functions in health and many important functions against development of malignancy through many of these mechanisms. It suppresses metastasis, it suppresses radiation-induced chromosome breakage. As we said, radiation-induced chromosome breakage can cause disease and it can cause cancer. The chromosome breakage might result in relocation of proto-oncogenes to new sites downstream of stronger promoters, وبالتالي يبدأ convert them to oncogenes. فده third mechanism زي ما قلنا هنا الانتي ميتاسيس mechanism بتشمل directly metastasis suppressor genes which suppress the effect of tissue metalloproteinases. Second anti-metastasis mechanism host immune response. Third anti-metastasis mechanism is the effect of melatonin. This is not a molecular origin of theoretical يعني significance. The cancer developed from one cell or from a region, from a field. Is it a field defect or a single cell defect? ممكن يعني مالناش تقدروا تقراه in detail في البيبر اللي على السايت على الانترنت. وبرضه الاخير دوت كنت يعني انا حطيته يعني كنت عاوز اقوله لكم بس لقيته ان هو يعني برضه of more theoretical basis فمش هنتعرض له evolutionary paradox اللي هو in short the cancer cell has selective advantages over normal cells and they try to create a new creature in their affected host لكن بينتهوا بالdeath of this host. ده اللي هو البارادوكس. Instead of creating the new creature they kill their host. وده هو معنى الايفولوشن. اللي عاوز يقراه يعني هيلاقيه في البيبر in detail شويه ممكن يعني يقراه لكن it is not of significance as regards our discussion regarding the genetics of oncogenesis. السايت ده في الميديكال جينيتكس هتلاقي انا حطيت المحاضره corrected هتلاقوها موجودة في PDF فورمات اللي هو الأونكوجينيتكس، ممكن تنزلوها وتقروها in detail، وهتلاقوا برضو الريفيو ارتكل عن الكانسر some genetic considerations، ممكن تنزلوه وتقروه، هيدوكوا details some theoretical details about some aspect of cancer ملهاش direct influence of the genetics of cancer. بس كده يعني. في أي أسئلة؟ انتوا نمتوا يا جماعة ولا صاحيين؟
أنا صاحية معرفة.
شكرا لحضرتك يا دكتور.
طيب.
صحيح يا دكتور.